I hope everyone's enjoying their lunch. My name is Madeline Greer. I want to thank you all for being here. I'm a staff scientist with Stapledine Instruments, Inc., and I'm going to give you a short talk today about our technology, the kinetic exclusion assay, in the era of AI and biologics innovation. So I have depicted here a schematic that shows the typical drug development process. Now, AI has shown potential in many of these different stages, but it's most advantageous early in the drug discovery process. So I have here denoted by yellow stars areas where AI has given us key advantages into making things more efficient, as well as providing us insight into target discovery, hit generation, lead identification, as well as predicting the outcomes of clinical trials. Now, as many of you know, bringing a drug to market is a very expensive, risky, and time-consuming process. It can take years and years, and today's market can cost upwards of $2 billion. So the reason researchers are looking into technologies such as generative AI is to not only help us understand potential targets and lead identification, but also to help us to find ways to shorten this timeline and reduce our costs. So in this figure, we see the potential cost savings in terms of millions of dollars in each of the drug discovery phases if researchers are able to reduce failure rates or cost by 20%, whichever is most impactful. Now, we see the biggest cost saving in the clinical trial phase as well as the lead optimization. And with so much cost savings on the line, it's no wonder researchers are trying to capitalize on this. However, to achieve this cost savings, it's important that the information that we're feeding into these AI algorithms is accurately characterized, and binding and functional parameters are being scrutinized. So here is a graph showing the amount of AI-derived molecules in clinical trials. Now, we see them spread out over all three phases with a dramatic increase in recent years, with one actually being launched in 2023. So next, I want to look at the types of molecules that are making up these numbers. So the types of AI-derived molecules vary widely, from targets to antibodies, vaccines, as well as repurposed molecules and others. The largest increase was The largest increase that we saw was in small molecules, which actually doubled from 2022 to 2023. Now, it's possible that this is because small molecules have relatively simple structures, but I think it lends more to the fact that there is a wealth of data available through databases that have been fed into these AI algorithms. Now, this presents an opportunity with more complex molecules, with more complex structures, that the more we know about them, the more data we can provide to provide for these programs to develop the most efficient therapeutics. So in addition to advancements in AI, we are also seeing biologic innovation that is transforming the therapeutic landscape. So today's drugs are no longer limited to the traditional canonical monoclonal antibody. We are now seeing things such as antibody fragments like scFvs and nanobodies, in addition to antibody drugs conjugates, functionalized liposomes, as well as multi-specific drugs that can target multiple epitopes simultaneously. Now, this is a really busy figure. It shows the number of different antibody formats by year from 1986 to 2022. And what I want to draw your attention to specifically is the bottom half of this graph. Traditional antibody structures still make up the largest class of these drugs. However, we are seeing an increase in other structures over time with our ADCs, our bispecifics, and other molecules. Now, as these constructs become more complex, accurate and sensitive characterization becomes increasingly challenging, but also critical to ensure functionality, safety, and efficacy. Now, each technology is going to approach characterizing these interactions in a different way. Surface-based technologies such as SBR, BLI, and LSA look at the ratio of kinetic constants, so K off over K on. I have here depicted a typical sensorgram where the increase we see here is the measurement of the K on, or the association rate, and then we directly measure the off rate, or the dissociation rate, and the KD can be calculated by using the equation shown here. So FACS, or fluorescence-associated cell sorting, tackles characterizing interactions specifically by looking the amount of bound antibody. So it specifically looks at concentrations versus the kinetic constants. And the data typically is presented as a sigmoidal curve where we have titrated antibody along the x-axis and signal on the y-axis. So as more of our antibody binds to that cell surface, the signal increases. And then we derive the KD from the EC50 or the 50% point shown here. So KinExA also looks at concentrations, but instead of looking at the bound on antibody, we specifically look at the unbound antibody concentration. A titration of the antigen is shown here along the x-axis, and we have percent free antibody along the y-axis. So as more and more antigen is present in each of our samples, there is less free antibody. And then the KD is determined by using exact solutions to these binding curves to fit the data. Now, for all these platforms, the dynamic affinity varies widely. While surface-based technologies like SPR, BLI, and LSA are used widely and effective across a broad range of affinities, they can face limitations when measuring extremely tight binding. KinExA excels in the tight affinity range. This makes it particularly valuable when measuring biologics that have been designed to have high specificity and really long dissociation rates. Next, I want to transition into looking specifically how KinExA measurements are conducted. So the typical KinExA method, antigen is titrated in the background of antibody. The first tube shown here has the highest amount of antigen, which is then titrated, and as antigen decreases, the antibody does not. These samples are allowed to reach equilibrium in solution and are then measured on the instrument. The binding curves are representative of how much free antibody is in each sample at equilibrium. At the highest level, the antigen has bound up the antibody almost 100%, and we can see that here represented on our y-axis, which is percent free antibody. At the bottom of our curve here, we see absolutely no free unbound antibody available. However, as antigen levels decrease in each of our samples, there's more and more free antibody available to bind, all the way up to 100% free. So how are we able to isolate this unbound fraction of antibody? In order to show you this, I want to narrow in on the flow cell that's hidden behind a door here on the instrument I have depicted in the corner. So first, we load in antigen-coded microbeads into our flow cell, and they are coated with the same antigen that's in each of our solutions. And I want to make it abundantly clear that this capture column is used solely as a method to probe the interaction that has already happened in solution. So we flow each of our samples over this capture column, where we capture any unbound antibody, and complexes are passed through the instrument. Next, we flow in an excess of fluorescent anti-antibody. And once we've washed away any excess here, what we are left with is a fluorescent signal that is proportional to the amount of unbound antibody in solution. So a couple other benefits of performing measurements with KinExA is that they are as close to physiologically relevant conditions as possible. You are not limited to a specific buffer. We can run buffers with a variety of different pHs, with different additives, as well as using whole human serum. We can also perform measurements from 4 degrees C all the way up to 37 degrees C. And samples are measured in solution with either titrated antigen or whole cells. So circling back to the drug discovery phase, this figure illustrates a typical problem with antibody discovery. We're starting with a large pool of candidates that are gradually narrowed down to a few targets. During this process, it's important to be able to differentiate various clones based on desired parameters. And affinity is often a parameter used to help aid in this selection. So with that information, potential leads can then be optimized to perform better than their reference molecules, and we can move down this funnel. So this figure shows the results of a screen for a large pool of antibodies that was generated by next-generation sequencing. So this screen was done with SPR, and we can see the on rate is on the y-axis and the off rate is on our x-axis here, and then affinity values can be seen up at the top, delineated by these red lines. Now in this screen, some antibodies with affinities below 100 picomolar have reached the detection limit of SPR, making it impossible to rank order them. on SPR, this is commonly seen as a flat line of disassociation. And the challenge then becomes how to effectively differentiate but prioritize these type binders. It would not be cost effective or efficient to individually screen each clone with KinExA, and so we were not traditionally thought of as a solution to this problem. However, I want to depict here a workflow that integrates the new KinExA single point screen. So the largest group of antibodies to weed out the weakest binders can still be done with surface-based technologies. However, when we need to move to a smaller pool of antibodies that have reached that detection limit, we can use the KinExA single-point screen that broadens the throughput we can use on KinExA, but maintains that ability, that sensitivity to differentiate between those sub-100 picomolar binders. And then we can measure that smallest pool of our lead candidates with the KinExA dual curve method. So KinExA plays a critical role in providing precise and reliable characterization of these biologics, ensuring that AI-driven decisions are based on accurate, sensitive, and reliable data. Together, these two powerful technologies can help make the most efficient therapeutic development. We've got a booth over at 205 and a poster over at 71. Please come talk to me for questions. Thank you.